Chapter 12, we're going to look at. So, mitosis was first observed in 1882 by German anatomist Walther Fleming. So, let's get down into how mitosis fits into the cell cycle. So, the two main parts of the cell cycle, one is M phase, which is cell division, mitosis and cytokinesis. And the other part is interphase, where the cell grows, DNA is copied, and we prepare for cell division. So let's look at that a little bit closely. Interphase, again, interphase is the time in between cell divisions. It alternates with M phase. This is about 90% of the cell cycle, okay? And it's divided up into three subphases. G1 phase, which is growth. S phase, which is DNA replication. And then G2 phase, which is prep for cell division. Okay. The cell does grow during all three phases, but chromosomes are only duplicated during S phase. So we normally set up the cell cycle like a clock. Okay. And if we start here, after cell division, the cell grows through G1, okay? Then as it enters into S phase, it will replicate its DNA. That's the first step it takes to prepare to divide. And then if it enters into G2, we prep here for M, for cell division. And then the cell enters M phase. It undergoes mitosis, followed by cytokinesis. Then we have two cells, and each of them start the cell cycle again. So let's narrow in on mitosis. Mitosis divided into 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. And cytokinesis overlaps with telophase, so much so that we mostly just call them telophase and cytokinesis because they sort of happen simultaneously. All right, so here is kind of a picture of all the phases together on one slide. So if you want to kind of screenshot this, I'll have a moment of silence while you screenshot it. There you go. We have interphase, prophase, prometaphase, metaphase, anaphase, and telophase, and cytokinesis. So I'm going to go through each of these in a little bit more detail, but that's kind of everything on one page because you should be able to look at the chromosomes and tell which phase you are in. All right, so G2 of interphase. So right before mitosis begins, okay, you have duplicated chromatin, duplicated chromosomes, but they're not condensed. They're very diffuse. The nuclear envelope is still intact, and it still looks pretty much just like a cell in interphase. Once you enter M phase, so from this point on we are in M phase. The first part of mitosis is prophase, and so you start to have a little bit of mitotic spindle forming, and the chromosomes, okay, begin to condense and become visible as chromosomes, whereas over here they weren't really visible as individual chromosomes. Okay, chromosomes consist of 2 sister chromatids, and they are joined at the centromere. Okay, as we move from prophase into prometaphase, the nuclear envelope begins to disintegrate, and as it disintegrates, you can have the mitotic spindle attach. So you will have microtubules from the mitotic spindle attached to the chromosomes, because that is what moves the chromosomes around. The spindle fibers move the chromosomes around, so they need to get attached to move the chromosomes into the formation for metaphase, which is next. Okay, I'm going to talk about what kinetochores are shortly. And yes, so then once the spindle fibers have moved the chromosomes into alignment at the center point of the equator of the cell, which we call the metaphase plate. Once they're all lined up, then the cell is in metaphase, and it's very ordered, very particular. We have a whole beautiful spindle, and they're all attached to the chromosomes so that it can further manipulate them as we go through the succeeding phases of mitosis. So once we have all the cells lined up, chromosomes lined up at metaphase, the cell can move into anaphase, where the sister chromatids separate. Sister chromatids separate. I'm writing too low. Sister chromatids separate. And you can see, so one copy of each chromosome is getting pulled towards the centrioles, okay, and the poles of the cell. Okay, once the chromosomes arrive at either side of the cell, we go into telophase. Now you see there are two separate areas where the chromosomes are. They form into the new nuclei. The nuclear envelope reforms, and we begin to have cytokinesis. A cleavage furrow forms. The cell pinches in, kind of like a drawstring or one of those cinch sack purses. There's a ring here that just kind of squeezes the center of the cell, and you're left with two cells. And again, they have identical, they're identical genetically. They have identical nuclei. Okay. And those are the phases of mitosis. You should also be able to recognize them in real cells. Okay. If you have a nucleus in a real cell that is just kind of diffusely stained, no distinct chromosomes, that'll be an interphase. Once you can start to see the DNA, you're moving into prophase, distinct chromosomes in prometaphase. Okay, metaphase is pretty easy to see. Everybody's lined up on the metaphase plate. Anaphase, you have the chromosomes being separating, sister chromatids separate and being pulled towards opposite cells. And telophase and cytokinesis, you see you have two separate nuclei now, or areas where the DNA is. Okay, I'm going to spend the rest of this screencast looking a little bit closer at the mitotic spindle. Okay, so because it's very important, this is what makes sure that all the chromosomes get where they need to be, that each daughter cell gets one copy and one copy only of each chromosome. And that's all up to the mitotic spindle. And it's made of microtubules, and it controls the chromosome movement during mitosis. Okay. In animal cells, the assembly of the spindle microtubules begins at the centrosome, which is called a MTOC, or microtubule organizing center. The centrosome contains two centrioles, and it replicates during interphase, and that will give you two centrosomes that can migrate to opposite ends of the cell during prophase and prometaphase. As the centrosomes migrate to the poles, what grows out of them is called an aster, or it's an array of short microtubules. Okay, extends from the centrosome. We're going to see a picture of this in a second. And so the spindle itself, the mitotic spindle, includes the centrosomes, the spindle microtubules, and the asters. So, well, we're still not ready for a look. Let's just take a quick look. Okay, so there's an aster. Okay, there's a centrosome, and these are my spindle microtubules. Okay, all right. The last thing I want to actually talk about here is how the microtubules attach to the chromosomes. And they attach at structures called kinetochores. Okay, So spindle microtubules during prometaphase, this is what defines prometaphase from prophase. Spindle microtubules attach at the kinetochores of the chromosomes and begin to move the chromosomes towards the metaphase plate. Kinetochores are protein complexes that are associated with the centromere, okay? And that's where the microtubules attach. Once you have the chromosomes all lined up at the metaphase plate, that's when you're ready for transitioning into the next phases of mitosis. So now let's have a look again. You have your aster, your centrosome, and then your spindle microtubules. And you can see here they are attached, some of them are attached to the kinetochores, which are on the centromeres of the microtubules. And here you have kinetochores, kind of this area right here. Okay, and it's a protein structure, and any microtubule that's attached to a kinetochore is called a kinetochore microtubule. There are also microtubules that are not attached to the kinetochore, and those are called non-kinetochore microtubules. And what they do is they basically hold the spindle fibers in place. They hold the entire structure in place. Okay. Once you have everything arranged at the metaphase plate, the cell transitions into anaphase when the sister chromatids kind of let go of each other. So they separate at their centromeres, and then they move along the kinetochore microtubules towards the opposite ends of the cell. And microtubules shorten by depolymerizing or falling apart at the ends that attach to the kinetochores. Okay, so the microtubule doesn't detach at this end. The microtubule shortens at this end, so it attaches at the kinetochore end. Okay, so and you'll see here, this is kind of a. I'm going to blow this up a little bit. Here's my chromosome here. Here's my kinetochore, which is right here, this complex. And you'll see there's little motor proteins. And this is like, you know, the guy who's walking along the microtubule in the Cell video, my favorite guy. He's a motor protein, and the same motor proteins are present in the kinetochore. And they walk the chromosome down the microtubule. As they do, the microtubule falls apart into tubulin subunits. Okay, and then that's what draws the chromosome towards, right? That's what draws the chromosomes towards the centromeres. Oh, sorry, towards the centrosomes. Okay, oh, now it's big. I've got to try to make it smaller. There we go. Oop, that's too small. Okay, and previously I mentioned the non-kinetochore microtubules. So they don't attach to chromosomes, but they are important in maintaining the shape of the mitotic spindle during mitosis. So they overlap each other and push against each other, elongating the cells and pushing the centrosomes apart. Okay. Once you're into telophase, there are genetically identical daughter nuclei, and then cytokinesis begins during sometimes as early as anaphase, but pretty much during telophase. And once that occurs, the spindle disassembles. Okay, and that's it for part 1. Okay, watch the next screencast for the remainder of mitosis.